when you swallow a pill, maybe to cure a headache or clear up a sinus infection, you probably assume its journey just ends right there in your stomach.
Right, yeah. You think the chemical transaction is complete, you feel better, and the story's over.
Exactly. But the truth is that pill is actually about to embark on this massive, invisible journey.
Oh, absolutely. A journey that doesn't just disappear.
Right. I mean, it's a journey that actually ends up fundamentally altering the gender of fish hundreds of miles away before eventually flowing right back out of your kitchen tap.
It is wild. The life cycle of our medicine is honestly one of the most profound blind spots in our modern infrastructure.
It really is.
I mean, we design these incredibly resilient chemical compounds to survive the human digestive tract, but we rarely consider what happens when that exact same resilience is unleashed on the open environment.
And that is exactly what we are unpacking in today's deep dive. So we're looking at this comprehensive, systematic review that tracks pharmaceutical compounds polluting 13 major Indian River ecosystems.
Yes, and the data here is just, wow, it's incredibly revealing.
So the mission here for you, the listener, is to map out the sheer scale of the drugs ending up in these water systems. We want to look at the biological mechanisms of how they are actively re-engineering aquatic life.
Right, and to really understand the boomerang effect, why this massive ecological disruption is, at its core, a direct threat to human health.
Exactly. And the source material for this, the data pool for this review is intensely rigorous, isn't it?
Oh, highly rigorous. The research teams, they started by casting this huge net over 427 articles.
Wow, okay, 400.
Yeah, but from there, they stripped away anything relying on mere estimates or theoretical modeling. They narrowed it all the way down to 22 highly specific studies.
You have 22.
Right, published over the last decade. And these are studies that provide quantitative, measurable concentrations of these drugs. So we are looking at verifiable chemistry in the water, not just projections.
Which is terrifying, honestly. Reading through this data, it feels like we are essentially prescribing highly potent medications to an entire ecosystem without a diagnosis.
That's a really good way to put it.
We've basically turned these major river systems into unintentional, completely unregulated pharmacies.
Unregulated is the key word there, yeah.
But to understand how bad it is, we really need to look at what scientists are actually finding in the water. If you walk down to a river and scoop up a glass of water, you don't see floating capsules of ibuprofen.
No, of course not. They are entirely invisible to the naked eye.
Right, so how do you find a microscopic needle in a river-sized haystack?
Well, you need a technology capable of sorting chemicals at a molecular level. And the primary analytical tool they highlight in the review is a two-step process.
Okay.
It's called liquid chromatography, coupled with mass spectrometry, or LCMS for short.
Okay, wait. I know chromatography involves separating things like when you put a wet marker on a coffee filter and the colors spread out in the water.
Yes, that is actually the perfect baseline to understand it.
Oh, really? So it's essentially a high-tech version of that.
Basically, yeah. In liquid chromatography, you take your complex river water sample and you force it through this specialized column packed with microscopic porous particles.
Okay, so pushing the water through a super fine filter.
Right. And different chemical molecules interact with those particles in different ways based on their polarity and their size.
Ah, so they move at different speeds.
Exactly. Some stick tightly and move really slowly, while others just slide right through. So out the other end, your complex soup is separated into these single ingredient streams.
That makes total sense. And then the mass spectrometry part kicks in to identify those individual ingredients.
That's exactly how it works. Once they're separated, the mass spectrometer essentially vaporizes those molecules.
Vaporizes them.
Yeah, it blasts them with electrons to give them an electrical charge, and then it measures their mass-to-charge ratio, because every pharmaceutical compound has this unique mass signature.
Like a barcode.
Exactly like an atomic barcode. The machine reads that barcode and can detect these compounds down to nanograms per liter.
Nanograms? Which sounds like an absurdly tiny amount, until you realize that pharmaceuticals are biologically active by design.
Right, they don't need to be in massive concentrations to trigger a massive biological response.
Exactly. So when they run this LCMS technology on these 13 rivers, what barcodes are popping up the most?
The inventory is staggering, honestly. The most prevalent culprits span several different categories of human consumption.
Like what?
Well, you have the anti-inflammatories and painkillers, so massive amounts of ibuprofen, diclofenac, and naproxen.
Wow, okay. Everyday stuff.
Yeah, but then you also have heavy-duty antibiotics, things like ciprofloxacin, norfloxacin, and sulfamethoxazole.
Those are the broad-spectrum drugs, right? The ones we use for severe infections.
Yes, the real heavy hitters. And beyond that, the rivers are saturated with chronic disease medications, particularly the diabetes drug metformin.
Metformin is everywhere in this study.
It really is. Plus, there are high concentrations of everyday stimulants like caffeine and antimicrobial agents like triclosan.
Criclosan, that's the active ingredient in antibacterial soaps and toothpaste.
Exactly. It just all goes down the drain.
Which brings up a really logical assumption, I think, that most people would make. Our domestic plumbing, hospital drains, it all flows into wastewater treatment plants or WWTPs.
In theory, yes.
Right, so why aren't the wastewater plants just filtering these chemicals out before they dump the water back into the rivers?
So, to understand the failure of the wastewater plants, we have to look at how conventional biological treatment actually works.
Okay.
The vast majority of these facilities rely on what's called activated sludge.
Activated sludge sounds lovely.
It's basically just cultivating massive vats of bacteria, and their entire job is to eat the organic waste in the water. They digest human waste, food scraps, nitrogen, all of that.
But I'm guessing a synthetic pharmaceutical molecule doesn't exactly look like an appetizing meal to a bacterium?
It doesn't look like food at all. Remember your earlier point about these drugs surviving the human stomach?
Yeah.
Pharmaceuticals are engineered with these highly complex synthetic chemical structures, often involving tightly bonded rings and halogens like fluorine or chlorine.
So they're built tough.
Very tough. They are quite literally designed with a sort of molecular armor to prevent enzymes from breaking them down prematurely in your body.
Oh, wow. So the very engineering that allows a pill to survive human stomach acid and reach our bloodstream,
Yeah.
that is the exact same engineering that makes it invincible to the bacteria in a wastewater treatment plant.
Exactly. The bacteria just pass on it. The synthetic bonds are completely foreign to them.
That's incredible. So it just flows right through.
Right through. The pharmaceutical molecules pass through the activated sludge virtually entirely intact.
And that's just the treated water.
Right. Add to that the fact that a lot of agricultural runoff, which is just full of veterinary antibiotics, bypasses municipal treatment plants completely.
And untreated sewage, too, I imagine.
Exactly, especially in certain rapidly developing areas.
Okay, so that definitely sets the stage for why they are in the water. But the data shows this isn't just a uniform blanket of pollution. There are major geographical disparities here.
Oh, huge disparity.
Well, let's look at the hotspots because the extremes in this study are just wild.
We really see that stark contrast when we look at rivers like the Swarna and the Netravati. The data there is surprisingly optimistic, actually.
Okay, so some good news.
Yeah, the concentrations of compounds like sulfamethoxazole were virtually non-existent there, posing a near zero potential risk.
But then you look at the Nag River and the numbers absolutely explode.
They do. The Nag River flows through Nagpur, which is this urban center experiencing explosive population density.
Right.
And in that river, researchers detected the diabetes drug metformin at 94.000 grams per liter.
Wait.
They also found the antibacterial agent triclosan at 74.300 grams per liter.
Let me stop you there. To put that 94 milligrams per liter in perspective, we just talked about the mass spectrometer needing to find nanograms, right? Billionths of a gram.
Yes.
94 milligrams is microscopic, sure. But in this context, it's macroscopic. If you had a standard water bottle from a vending machine, you'd almost be able to see the physical weight of the drug in the water.
It is an unbelievably high concentration for an open waterway.
But as shocking as the Nag River is, the Sursa River data is what really stopped me in my tracks.
Ah, the Sursa. Yeah, it is the ultimate cautionary tale of industrial overlap.
What were the numbers there again?
The review logged the antibiotic norfloxacin at an incredible 277 milligrams per liter.
That is just insane.
And ciprofloxacin was at 203 milligrams per liter.
That concentration is so high, it fundamentally stops being a river and starts becoming a vat of medical intervention.
It basically is a diluted chemical soup at that point.
How does an open body of water reach nearly 300 milligrams per liter of a potent antibiotic? That can't just be from human urine passing through a wastewater plant, right?
Right. No, it isn't just human excretion, not at all. The Sursa River is heavily impacted by industrial cluster zones.
Oh, okay.
It basically functions as the receiving body for the effluent coming out of centralized wastewater treatment plants that serve as actual pharmaceutical manufacturing hubs.
Oh, so this is the runoff from the factory floor where the drugs are actually being synthesized and packaged.
Precisely. You have the raw, active pharmaceutical ingredients being discharged in highly concentrated volumes.
Which just overwhelms everything.
Constantly. It completely overwhelms whatever limited chemical filtration the plants might attempt.
At those concentrations, the obvious question is what this is doing to aquatic life. Because fish don't metabolize drugs the way humans do, and they are submerged in this soup 24/7.
Exactly. And the researchers actually quantify this threat using a specific metric. It's called the ecological risk quotient, or RQ.
RQ, okay.
It is a ratio. It compares the measured environmental concentration, so what they actually pulled from the water, against the predicted no-effect concentration.
Got it. So it's what is actually there, divided by the absolute maximum amount scientists believe the ecosystem can handle without taking damage.
Correct. And the safety threshold is exactly 1.
Okay, so over 1 is bad.
If the RQ is above 1, the water is toxic to the organisms living in it.
And in the Sursa River, the RQ for that antibiotic, norfloxacin, was 120.43.
Right.
It is 120 times the maximum safe ecological limit.
It's astronomical. And the painkiller diclofenac breached the RQ threshold of 1 in the Ahar River as well.
Which is where we really need to look at the specific biological mechanisms of the damage. Like, what is actually happening to the fish?
Let's take diclofenac as an example. When fish are continuously exposed to this painkiller, it induces profound oxidative stress.
And that specifically targets their kidneys.
Right. Specifically the kidneys.
The oxidative stress is one of those terms that gets thrown around a lot in health circles,
Yeah.
get your antioxidants and all that. But what is actually happening inside the fish's kidney cells?
Well, normally cellular metabolism naturally produces free radicals. These are basically unstable oxygen molecules that want to rip electrons away from other structures.
Like rust, kind of?
Basically. But our bodies and fish bodies naturally produce an enzyme called superoxide dismutase.
Superoxide dismutase.
Yes. And it acts as a shield, neutralizing those free radicals before they can cause any harm.
So it's a built-in antioxidant defense system right inside the cells.
Exactly. But what diclofenac does is it aggressively alters and suppresses the production of that superoxide dismutase in the fish.
Oh, wow. So the shield goes down.
The shield goes down, and without that enzymatic shield, the free radicals just run rampant.
What do?
They start ripping apart the lipid membranes of the kidney cells, which leads to complete tissue degradation and eventual organ failure.
Man. So the painkiller is systematically dismantling the fish's cellular defense mechanism, basically allowing its own metabolism to tear its kidneys apart.
It is brutal.
That is awful. But the review points to another chemical interaction that might be even more structurally devastating to the ecosystem, right? The endocrine disrupting chemicals.
Yes, the EDCs. And the diabetes drug metformin is a prime example in this study.
How does that work?
Well, EDCs don't necessarily cause immediate cellular death like oxidative stress does. Instead, they hijack the fish's biological communication networks.
They're hormones.
Exactly. Metformin interferes with the endocrine axis. The data shows it can actually upregulate the expression of an enzyme called aromatase.
Okay, and what does aromatase do?
In a fish's body, aromatase is responsible for converting male hormones, the androgens, into female hormones, the estrogens.
Wait, so by upregulating that enzyme, the diabetes drug is forcing the male fish to chemically convert their own testosterone into estrogen.
Yes. It creates such a profound hormonal imbalance that it literally induces male to female sex changes in wild fish populations.
That is just mind-blowing.
You end up with rivers where the sex ratio is drastically skewed.
And if you skew the sex ratio, the population crashes.
Completely.
Because they can't reproduce. And if the fish population crashes, the predatory birds starve, the algae they usually eat overgrows, and the entire food web just collapses.
It's a systemic unraveling. And it's caused by a drug meant to just regulate human blood sugar.
Which brings us to the most terrifying part of this entire analysis, honestly, the boomerang effect.
Right, because humans rely on these rivers.
Exactly. This water eventually makes its way into drinking supplies, into the agricultural soil used to grow food, and into the fish that humans pull out and eat.
So we have to look at human toxicity. And to do that, the study uses something called the risk quotient for human health, or the RQH.
Okay, so like ecological RQ, but for us.
It works very similarly, yeah.
Yeah.
But it incorporates the acceptable daily intake, or ADI, and that factors in human body weight and daily water consumption.
Right. And the numbers here are what I really want to push back on, or at least try to contextualize for the listener.
Okay.
Because the data says that for adults, In major lifeline rivers like the Kaveri, the Yamuna, and the Ganga, the RQH values for various drugs far exceeded the safety threshold of 1.
They did, yes.
In the Kaveri River, the human risk quotient for naproxen was 520.83.
520 times the acceptable risk limit.
But here is my question. If people living along the Kaveri are drinking water that is 500 times the toxic limit for an anti-inflammatory drug, why aren't we seeing people dropping dead on the riverbanks?
That is a very fair point.
Right. Like, if the toxicity is that high, where is the acute health crisis?
That is a brilliant distinction to make, and it really comes down to the difference between acute toxicity and chronic sublethal toxicity.
Okay, explain that.
The RQH isn't measuring the threshold for a lethal overdose. It's not, poison control.
Right.
It is measuring the threshold where the biological safety mechanisms of the human body begin to fail under constant daily stress.
Ah. So it's not a single dose of poison, it's more like a slow, agonizing marathon of organ strain.
Exactly. When you consume 500 times the acceptable daily limit of naproxen every single day for years, you are subjecting your gastrointestinal tract to constant irritation.
Which causes ulcers.
Yes, severe ulcers and GI bleeding over time. You're basically forcing your kidneys to perpetually filter a synthetic compound they were never meant to process at that volume.
Which leads to long-term renal impairment.
Precisely.
And the study highlights that this chronic exposure is exponentially more dangerous for children.
Right.
Okay.
Vastly more dangerous.
Now, the obvious assumption is just that children weigh less. The study uses a 15-kilogram baseline for kids versus 70 kilograms for adults. But a smaller body mass can't be the only reason their vulnerability is so much higher.
You're right. It's not just about mass. It's about biological momentum.
What do you mean by that?
Children actually consume a significantly higher volume of water relative to their body weight than adults do.
Oh, I hadn't thought of that.
And more importantly, their organ systems are actively developing. Their cell division rate is incredibly high.
Right, so if you introduce an endocrine-disrupting chemical, like metformin, into a child's system right as they are entering puberty, you aren't just altering a stable adult system.
No, you are corrupting the foundational blueprint.
Wow.
The EDCs interfere with the developmental baselines. Disrupting hormones during those critical developmental windows can lead to permanent reproductive and metabolic disorders.
Disorders that just cascade through the rest of their lives.
Exactly. It's a lifelong impact.
But as devastating as the direct organ and hormone damage is, the review isolates one specific outcome as the greatest existential threat to global public health.
Antimicrobial resistance.
Yes, the creation of superbugs. This is where the antibiotic concentrations in rivers like the Sursa become a global emergency.
It really is an emergency.
Let's break down the mechanism here, because a lot of people might think, well, if there are massive amounts of antibiotics in the water, shouldn't that just kill all the bacteria and make the water cleaner?
That is the intuition, sure, but it's entirely backwards.
How's
The concentration of antibiotics in these rivers is incredibly high for an open ecosystem, but it is still sublethal for a lot of bacteria.
this lethal?
Yeah, it's what microbiologists call a subminimum inhibitory concentration. It's not enough to instantly rupture the bacterial cell wall and kill the colony.
So it's basically just a non-lethal stress test.
A perfect way to describe it. By exposing the bacteria to sublethal doses of ciprofloxacin and norfloxacin, you trigger a profound stress response in the pathogens.
And they fight back.
Right. Under chemical stress, bacteria accelerate their mutation rates. And even more dangerous than that, they engage in horizontal gene transfer.
Horizontal gene transfer. Wait, that's when bacteria literally swap DNA with each other, right?
Yes. If one bacterium randomly develops a genetic mutation that allows it to pump the antibiotic back out of its cell, it can package that resistance gene into a small loop of DNA.
That's a plasmid, right?
Exactly, a plasmid. And then it physically hands it over to a neighboring bacterium.
Just hands it over.
Just passes it along. Even if they're a completely different species of bacteria.
So the river acts as this giant fluid boot camp where pathogens are constantly sparring with our best medicines and then trading the blueprints for how to defeat them.
And that leads to a terrifying public health reality.
Because people drink that water.
The rivers in this study are heavily relied upon by millions of people. And waterborne illnesses, like severe diarrhea and cholera, are already endemic in many of these regions.
So when those people get sick,
When those pathogens inevitably infect the human population, the standard antibiotics prescribed by doctors simply bounce off them. The drugs have been rendered entirely useless.
By the very water that transmitted the disease in the first place.
Exactly.
It's a perfectly closed loop of systemic failure. You take the antibiotic to cure the disease, you excrete it into the river trains the next generation of the disease to be immune to the antibiotic, and then it flows right back into your drinking glass.
Which is why the authors of this systematic review tie this local data directly to the United Nations Sustainable Development Goals.
Right, because it's not just an isolated issue.
No, this isn't just an Indian infrastructure problem. This is Goal 6, clean water and sanitation, violently clashing with Goal 12, responsible consumption and production.
I mean, our entire model of pharmaceutical consumption is built on a linear assumption in a circular world. We assume the toilet flush is just the end of the line.
But the line is a circle, and the review clearly calls for a radical overhaul.
What kind of overhaul?
Well, not just advanced wastewater technologies capable of chemically dismantling these stable molecular rings. We also need strict, enforceable regulations on the pharmaceutical industry's own disposal practices.
Because the industrial runoff creating those 200 milligram per liter hotspots of norfloxacin is entirely preventable, right?
Entirely preventable with proper closed-loop manufacturing regulations. The technology exists to treat it on-site. It simply isn't being mandated or enforced.
As we wrap up this deep dive, it really shifts how you have to view your own body's relationship with the environment.
It really does.
We have spent centuries treating medicine as a localized internal event. The pill goes into your body, fixes your biology, and that's it. We draw a hard line between the inside of our bodies and the outside world.
But this research proves that boundary doesn't exist. Your biology is porous, and the environment is just an extension of your own bloodstream.
It leaves you with a deeply unsettling question to ponder, especially as we see these resistance genes multiplying.
Definitely.
If our water supply is becoming a permanent low-dose cocktail of synthetic chemicals and antibiotics, will human biology eventually have to adapt to it?
That's the big question.
Right. Are we pushing our own gut microbiome toward a future where it has to evolve to synthesize or even depend on this synthetic environment just to survive?
It's a very real possibility.
It's a heavy thought for the next time you pour a glass of tap water to wash down a headache.